Our next speaker for this afternoon is Ignacio Vargas from Pontificia Universidad Católica in Chile. He's going to talk to us about changes in microbial community structure from natural extreme environments.
Thank you. Thank you for the opportunity to present our work here in this conference. My name is Ignacio Vargas. I'm from the Pontificia Universidad Católica de Chile. As the title of my presentation is Shift in Bacterial Community Structure from Extreme Environments Following a BES Incubation. This work was made in collaboration with Jay Regan at Penn State University. So the motivation of our research is the opportunity to find new electrochemically active bacteria from contaminated and extreme environments, as you can see here in the pictures. In particular, the exploration of these metal-rich environments for lithotrophic bacteria able to use sulfur, iron, or arsenic as electron donor and oxygen selector receptor presents an opportunity to develop low-cost biocathodes. So today, I'm going to present the results from two sites. The first one is an acid mine drainage affected area in central Pennsylvania. And the second one is an arsenic-rich environment in the north part of Chile. It's a natural contamination there because it's close to the volcanoes, a hot spring in this area. So the objective of our research in general is to use a bioelectrochemical system as an enrichment strategy for lithotrophic bacteria able to use iron arsenic as an electron donor. They can oxidize those minerals, and also they should present some electrochemical activity. So what we did was we take the samples, soils from sediments from those environments, and we set up different types of reactors in the laboratory using those sediments as inoculum, and then we collect the biocathode biofilms, and we use pyrosequence 16S pyrosequencing to analyze the communities and compare the communities with the community from those environments. So regarding this first site, it's an acid mine drainage affected soil. So as you expect, the pH was low, was lower than 3 actually. Also high iron content, but also the chemistry of arsenic and sulfur in the environment. So what we did here was take those sediments to construct Winogradsky columns used as bioelectrochemical systems. 3 different setups with different times long-term operation. And basically, we insert the anodes on the sediments and use the organic matter on the sediments and let graphite brushes on the upper part, the water part of the column as a cathode. And after time, we collect the cathodes and use those cathodes in a electrochemical cell and measure using linear strip ultrametry changes on the cathodic reaction of the cathodes. So, and for all the setups, we observe a difference on the catalysis compared with the this is the control that is just an abiotic control of the cathode. And then we kill the bacteria after gamma irradiation, and we observe a decrease on this catalysis. This is the dark dots that you can see there. So what we believe that there's a kind of catalysis, a microbial catalysis of the microorganisms growing over the fibers of the brushes. This is a fiber of the brushes, small biofilm growing there. So that could be the responsible for this change on the cathode performance. So what we did next, we used this pyrosequencing to analyze the community. This is the community structure of the field. The pyrosequencing revealed 21 phyla on this environment, mainly dominated by protobacteria, actinobacteria, acidobacteria, chloroflexia, and that as the main members of the community. But after incubation, the community shift. This is for the long term, the 10-month reactor. So the community of the cathode change to protobacteria and active bacteria, but also firmicutes appear in the biocathodes. And same for the other reactor. We observe protobacteria, actinobacteria, and also firmicutes. The same for the third one. So we ran these experiments with replicates, and also we did replicates of the 16A biosequencing. So we, at the end of these experiments, we changed the community from 21 phyla to, I guess, was 13 for those reactors, but mainly dominated by 3 or 4. So one interesting aspect of this experiment was that the bacteria related to the iron and sulfur chemistry were enriched after the incubation, and especially for iron and sulfur oxidizers. That's interesting because we observed genera from the species from the genera Theobacillus, that there is some reference about that, but almost according to our knowledge, not too much information about the Leptospirillum sulfobacillum. So that is an indication that probably is good candidates to be used in a biocathode or as a biocathodic microbes. Also for the iron and sulfur reducers, we also observe changes. The abundance on the field was very low, actually lower than the detection level on the field, but after this incubation, they increase the frequencies on the biocathode. So that's another good output of this reactor. So for this first bunch of experiments, we learned that we can use a bioelectrochemical system as an enrichment of lithotrophic bacteria, or at least to enlighten some new candidates to be used to more deep studies. So then we did this second project in the north of Chile. This is an arsenic-rich environment. It's a natural contamination there. It's just by a volcano, so hot spring, low pH again, not that low. It's a pH about 6. High arsenic concentration in the environment is concentration in the water, and in the precipitates, the water is about 1 milligrams per liter. It's 100 times the maximum that we tolerate in drinking water. So it's a very arsenic content environment, but also the chemistry of iron and sulfur is present. So this is a kind of similar environment that the site 1. What we did here, we checked samples from these sediments, and we used to inoculate two types of reactors. The same column reactor, so used as a sediment microbial fuel cell, and typical cube reactor is a single chamber air cathode microbial fuel cells. So about the columns, they were operated over 15 months. In the laboratory, we did in duplicates. We test. We insert the anodes. We use graphite and titanium anodes, and they were inserted in the sediments. And we use carbon-filled cathodes. This is the cathode that you can see here. It's the upper part of the column. And after that time, we take the cathodes and we again measured through LSB some changes respect to the abiotic control. And one of the things that we observed then using SEM microscopy is a microbe, just a little bit, not a big biofilm, but some cells growing on those cathodes, those carbon-filled cathodes. So that could be a suggestion of some microbial activity on the surfaces if you add the LSB results. So regarding the community, so the community from the field was composed mainly by 12 phyla, was more diverse than we expect at the beginning due to these extreme conditions. But after incubation, the community again shift to mostly protobacteria, and in the case of this reactor, Bacteroidetes appear as an important part of the community. It's in the screen here is yellow, but you can see Bacteroidetes here. And also actinobacteria has part of the community. But with the cubic reactor, they were operated as a batch cycle mode for 9 months, treating an acid medium with a pH about 3.8. And we did, as usual, performance. We collect current. But also at the end of the experiment, we take attention on the thick biocathode that we obtain on these air cathodes. And we did LSB again, observed this peak, and we extract the biofilm, extract DNA, and again compare with the source of the inoculum. And here again, this is the site, it's the different samples, but the same site, so it's mainly the same composition of the environment. And again, after this incubation, we observe a change in the community, a shift to protobacteria, bacteroidetes, actinobacteria, as well as the columns. But here the firmicutes appear again. As for us was interesting, since the change, of course, is the configuration, but mostly is the pH. We have a low pH here, and as well as in the site 1, where the firmicutes were part of the community. So maybe there is something in these biocathodes related to firmicutes and low pH conditions that we don't know, but it was just an observation. So to confirm these differences, this shift on the community for site 2, we did this principal correspondence analysis on the samples, and we have, you can see here, the 4 field samples are all together, and after incubation, they shift depending on the reactor and also the operation conditions to very, very different communities. So that, for us, was very interesting because we're trying to go up to the mountains and get a very cool sample, but at the end, the communities converge. Of course, the environment makes some boundaries, and in our case, they give us microorganisms related to the chemistry of arsenic, iron, and sulfur. So the next step was to get some isolates. So for this column, in the cathode of this column in site 2, we obtain 10 isolates, as you can see, the red ones in this tree, but from the environment we just obtained 2. That was, so far with the capacities of our lab, the best what we did at that time, but as first output, at least this enrichment helped us to obtain isolates from this inoculum. So I want to enlighten this cluster here because we obtained same isolates from the biocathode than from the site, and those were related to the arsenic cycle. Actually, we target for a gene of arsenic oxidation, a Rubisco gene for autotrophic, and they grow in a medium using just arsenic as electron donor. So we think that those guys are arsenic oxidizing bacteria, and the enrichment in this case, they don't show too much, but they give us a clue that maybe if they're part of the biofilm and at the cathode, they could have some electrochemically activity. So we did again an LSB with using electrochemical cells with those isolates, and we observed this current production, this nice peak. That could be an indication of some activity of those isolates, arsenic oxidizing isolate. So maybe we're just changing the arsenic for the cathode as electron donor. Also, we observed that this peak was related with the biomass. So we had different concentration of biomass to the reactor, and we observed different performance. So, but also this enrichment gives us the chance to observe other members not really related with the chemistry of the environment. That's the case for instance, the Pseudomonas fluorescens, that his metabolism is not really related with the arsenic chemistry, but it was present in the inoculum. So through this enrichment, this kind of the second output of this strategy, we can collect this potential new strain, or maybe, I don't know, we don't know if it's different species, but closely related to the Pseudomonas fluorescens. And now we're testing for its electrochemical activity. So as a general conclusions of this work is the incubation of this BES incubation imposed some restrictions that change the community structure from the environment to the biocathodes. The community depends on the operation condition of the reactor. For sure, that could require more attention and investigation. In our case, protobacteria, firmicutes, and bacteroidetes emerge as abundant phyla on our biocathodes for both sites. Also, our results, we believe, give us a first clue or initial indication for new electrochemically active bacteria potentially be used in the biocathode. So now we have a bunch of isolate that could be used for more deeper studies, applying different potentials and testing the changes on the metabolic activity running in an electrochemical system. So finally, I want to invite you to a poster that we have related to the acid mine drainage, but now using an MFC as a treatment strategy to change the pH of these environments. And also I want to thank our funding agencies in Chile, my group, especially Claudia Rojas. She is a former postdoc of my group. Now she's working with Jay, and she was leading this project. And of course, thanks to you for your attention.
Questions for Ignacio?
Thank you. So do you have any idea in your LSV experiment, some biotic control, there are still some current coming out. So do you know what happened there?
We just observe current at the very low potential, but we will list some related to the hydrogen that is produced in the cell, but it's a more negative potential that we observe for the biocathode reactors.
Any other questions?
Was it just me, or did one of your LSVs have a midpoint potential that was much more positive?
Yeah, they were different. I mean, they show different behaviors.
But most of your LSVs were negative.
Yeah.
And that one was shifted very positive.
Yeah.
And I'm just curious if you did these were pyrosequencing, right?
Yeah.
And so you're just zoomed out at the level, at the class, at proteobacteria. Do
Yeah.
you know, or even beyond that, sorry, do you know anything about the diversity of the proteobacteria that you're seeing?
Yeah,
and
now
if
we're
it's related to the midpoint potential?
Yeah, well, now we're looking on that, but it's mostly related to iron oxidizers. We got some Acidobacillus, Leptospirillum, and Sulfobacillus, so they could be responsible for this, more positive potential. But it's part of the work in progress. We don't know yet.
Did you measure any rate of iron oxidation or arsenic oxidation, and also any changes in pH during the runs?
Related to the pH was very stable for both sites in the reactors. For columns, they maintained, in the case of site 1, a low pH, really about 2.7, and the second one was about 6.3, but was stable. But now we're measuring the arsenic for the site 2. For the iron, we didn't measure at that time. So we don't have the information about changes on the iron chemistry during the operation. But I agree with you, it's something important.
Let's thank Ignacio. So it's time for a coffee break. We'll reconvene in 20 minutes. Speakers for the next session, please make sure your talks are uploaded. Thank you.